Good morning. I'm Bill Fazell. I'm a kidney doctor, biomedical engineer, and associate professor of medicine at Vanderbilt University Medical Center here in Nashville, Tennessee. This is a rebroadcast of a renal grand rounds I presented recently here on campus. And the message I'd like you to take away from this is that a bioartificial kidney is much closer than it appears. Since this is a continuing medical education event, I need to disclose that I'm the co-founder and member of a company incubating some of these technologies for renal replacement therapy, and I'm the inventor of some technologies we'll discuss today that someday might be used for clinical care.
Over the next 45-50 minutes, you're going to learn the performance objectives that we selected for an implantable artificial kidney, and then how those performance objectives informed our design strategy. We're then going to take a dive into the technology behind the hemofiltration membrane. That was the first innovative technology that forms my and our contribution to the field. And then we're going to talk about renal tubule cells, the living human cells that accomplish key functions in an artificial kidney. That's our second contribution to the field. And then I'm going to close with sort of the trailer, the teaser about future directions in testing the hemofilter in the bioreactor. I'm going to start with the thank yous. David Humes is a pioneering nephrologist and biomedical engineer at University of Michigan. And 25 years ago, when I was a second-year medical resident, talking about how I was going to make artificial kidneys out of silicon chips, he was one of the very few individuals in America who had the vision to listen to what I was saying, foresee the possibilities of bringing a new technology into a stagnant field. And he gave me a job when nobody else would really even give me the time of day. The other individual is Shuvo Roy, who's a biomedical engineer presently at University of California, San Francisco. He was then at Case Western Reserve University, where I was finishing my residency. I had the good fortune to meet him when he was initiating his lab at the Cleveland Clinic. And I told him that I was looking for a collaborator with experience in biomedical nanotechnology. And that was the birth of a 25-year collaboration. And I could not have done any of this without him and without David Humes. My lab at Vanderbilt is relatively small. Rachel Evans, the cell culture technician, and is a very wise woman beyond her years. Every time I ask her to perform an experiment, she actually performs a shadow experiment, duplicating the experiment, but knowing that she has a fallback in case I change my mind or my initial idea was a bad one. Hal Love is a staff scientist with PhD, and he's been responsible for a lot of the work that we've done, experimenting with the scaffolds in which we grow cells. Kuniko Hunter is a gifted biomedical engineering graduate student who has taken a deep dive into the mechanisms by which we're altering the performance of these cells in the dish. So when we think about what are our objectives for a bioartificial organ, it helps to understand what functions the healthy kidney performs. And these are a wide range of functions, so at least a dozen things that kidneys do for you. They regulate how much fluid you have, they regulate electrolytes, they regulate your osmotic balance, they get rid of waste products, they defend your body's acid-base balance, how much you're like vinegar versus how much you're like baking soda. They regulate the production of red cells in your bone marrow. They do a number of things. Our experience over 50 years taking care of patients with end-stage kidney disease has revealed that a lot of these functions can be duplicated with oral medicines or intravenous medicine. So we use sorbents like Veltassa or Renvela to control dietary absorption of some of the waste products that are hard for a kidney to get rid of. We can give injections of erythropoietin to promote red blood cell production by bone marrow. So there's actually very few functions that we need to focus on. And the one that's essential that we'll never be able to ignore is getting rid of waste products. And so that's where we focus. Your body makes a large amount of waste products from dietary intake of nutrients. So maybe 10 milligrams a minute of urea nitrogen, for example. And so this gives rise to an unavoidable tension between the need to remove a lot of waste products over the course of a day and the need to maintain those waste products at very low concentrations in your blood to prevent you from becoming intoxicated with the waste. And so if you've got to remove a large amount of something that's scarce, you have to process a lot of blood. Hemodialysis, which is the default treatment for renal failure in the United States today, because kidneys for kidney transplant are so scarce. Hemodialysis does this in 3 sessions a week, where the patient is connected to a machine, blood circulates over a filter, and waste products diffuse from a high concentration in blood to a low concentration in dialysate. And so you can reposition this as the idea that the dialysate, the hundreds of liters of salt water involved in every dialysis session, in essence, dilutes the waste from blood into this huge volume. On the other hand, kidneys do the opposite. Kidneys concentrate dilute wastes from your blood into a small concentrated volume of fluid called urine. And the loss of that fluid, the excretion of that fluid, can easily be balanced by oral intake. And since you can't carry a swimming pool around on your back, hemodialysis as a therapeutic strategy for an implantable artificial organ stops right at the get-go. You can't carry around a swimming pool on your back, and sorbents, etc., really involve disposables, involve materials that will be consumed and discarded. And that's a fundamental barrier to implantation, because you don't want to be taking something in and out past somebody's skin. So led us 20 years ago to decide that we had to embark on a challenge of concentrating waste products out of the blood into a small amount of fluid that we could get rid of and allow the patient to balance that fluid loss by oral intake. And there's a lot of ways to concentrate. So in the upper left hand, you see people making table salt out of seawater through evaporation. On the lower left-hand side, if you had just a single waste product that you wanted to get rid of out of water, you could maybe devise some strategy to precipitate that waste from solution to a solid and then get rid of the solid. On the lower right-hand corner, if you had only a few to get rid of, you could consider column chromatography and fraction collection, and just separate the constituents of plasma water and get rid of the fractions that you don't need. Then finally, in the upper right-hand corner, there's solid phase extraction, very commonly used in chemical chromatography and laboratory analysis, less commonly used in kidney failure, but what you see there is one of the first dialysis machines called the Ready machine, and the white cone on the left is a reactor of sorbents that absorb waste products out of the blood. Your kidneys do none of these things. Your kidneys work through the active transport of waste products by an epithelium. And what do I mean? Each one of your kidneys has a million little subunits. And in aggregate, about 1,700 liters of blood a day flow through the kidneys. Each one of those million subunits has a filter and takes about 8% of the fluid volume of blood passing through it and filters it, keeping antibodies and clotting factors and platelets and red blood cells in the bloodstream where they belong, but allowing a watery ultrafiltrated plasma to pass out of the bloodstream. And that in aggregate is 120, 140 liters a day, a much larger volume of fluid than you could replace by drinking. And so a second part of each one of these subunits of the kidney, called the tubule, is this lawn of epithelial cells that I've shown here with these little different colored bodies. And they reabsorb, they pump salt, water, amino acids, sugars back from the ultrafiltrate into the bloodstream. What they don't reabsorb are waste products. In fact, they will actively excrete waste into this fluid that's going to become urine. And they concentrate that 120, 140 liters a day down to the 1.7, 2 liters a day of urine that you pass into the toilet bowl and out to the Cumberland River. So 1,700 liters a day of blood becomes 1.7 liters a day of waste. And this is powered, the filtration is powered by cardiac perfusion pressure from the patient's heart. And the concentration mechanism is powered by the cellular energy of ATP from the body's metabolism. And that leads us to our strategy for waste removal. We set out to design a very high efficiency hemofiltration membrane that could be powered by the human heart alone, that would give rise to a clinically relevant volume of fluid every day, that we could then pass over a lawn of cultured epithelial cells that, using the cellular energy of ATP, would actively separate metabolically valuable substrates and return them to the bloodstream and excrete waste products into urine. So once we have this architecture in our mind, how much of it do we have to do? When you prescribe a medicine, you choose a dose. So setting out 20 years ago, we asked ourselves, is there some natural rate of filtration that would come close to balancing dietary intake? And so when you look at how much of each mineral you absorb a day and what the concentration is in circulating blood, you can calculate kind of how much of that you'd have to get rid of every day to balance dietary intake. And when you look at potassium, you can calculate about 20 liters a day. And remarkably, when you look at phosphorus, one of the other critical minerals that accumulates in renal failure, you also come up with about 20 liters a day. Calcium is a little bit harder, maybe it's a little less, 10 liters a day, which suggests that if we target 20 liters a day, we'll have to be careful to make sure we supplement dietary calcium. Nitrogen is a little more problematic. If we get rid of 10, 14 grams a day and we keep it at a plasma level that's normal, 10 milligrams per deciliter or 100 milligrams per liter, that implies we have to filter 100 liters a day, just like healthy kidneys do. However, urea nitrogen itself is not toxic. It's a surrogate for waste products in the blood. In fact, it's even an FDA-approved drug. I don't think any kidney doctor in the world would argue that a BUN of 50 milligrams per deciliter, 500 milligrams per liter, is dangerous or toxic. And if we accept that threshold, then we again converge on this 20 liters a day number. So we had an architecture and we had a dose that we wanted to achieve. And so with all of the confidence of a little boy tying on a cape that he got out of the washing machine and leaping off the garage roof, we set out to see if our ideas could fly. I'm going to start talking about our hemofiltration membrane. When you think about a dialysis machine and a dialysis unit, you see pumps. Every single dialysis machine you've seen has a pump. And it needs this pump driven by an electric motor that's plugged into a wall outlet because the hollow fibers of the dialyzer require a huge pressure to push the viscous colloid of blood through each hollow fiber. And the pores in this dialyzer are these irregular round apertures of varying size. And this is totally different than the human kidney that we see on the right, where the filtration structures are these very regularly sized, slot-shaped openings between adjacent cell processes. And so we asked Andrew Zidney, the chairman of chemical engineering at Penn State and the author of the textbook about ultrafiltration, microfiltration, if there was a physical reason for this. And so in one of our early publications, we showed that from first principles, a membrane composed of slot-shaped pores, shown here in the solid line, that are all the same size in the rightmost line, outperforms a membrane of round pores and outperforms a membrane where the pores have different sizes. So for any given ability to reject a large molecule, the membrane of slot-shaped pores lets through more water. And for any given water permeability, a membrane of slot-shaped pores significantly outperforms a membrane of round pores. So it appears that in evolving a glomerular slit diaphragm, nature optimized this trade-off between permeability and selectivity that's part of every filtration process. So armed with that, we proceeded to borrow a technology from the microelectronics industry. The technology is called silicon micromachining, and we used it to make these flat sheet membranes where the pores through the membrane can be seen as this crack right here between this piece of silicon and this piece of silicon. And when you break the membrane in half and you look at it in cross section, you can see that each one of these pores is the same size. It's an elongated slot going into the plane of the projection screen. It's a very smooth wall, nothing's going to get hung up there. And each one of these pores is ever so slightly smaller than serum albumin, so that it will retain albumin and clotting factors and cells in the blood, but let through salt water. So after prototyping this and testing it on the bench, we went on to implant it in experimental animals. And very quickly, we ran into a problem. I designed this very crude holder for the membrane that takes the round flow of blood vessels, transforms it into the sheet flow appropriate to our flat sheet membrane, then returns it to the round flow of a blood vessel. And every single time it clotted, and it clotted on the inside radius of the outlet. And it formed this white clot that was rich in platelets that eventually began to occlude the channel and lead to a coagulation clot behind it. And so this concerned us. And so we used a technology borrowed from aviation, borrowed from the artificial heart program, called computational fluid dynamics, to try to understand what was happening in blood. And so this is the shape of the channel in the plastic device I just showed you. And we play a movie showing the fluid shear stress inside the device. You can see that on the inside radius here, there's little areas that never wash out, that always have low shear stress. And so a platelet that happens to land there isn't going to get washed out, and so they accumulate. And that leads to the clotting. We changed our design in a couple of ways. We developed a helical inlet channel that actually mimicked the architecture of many blood vessels in the body. Because when blood turns a corner, or any liquid turns a corner, the high velocity jet in the center of Poiseuille flow hits the wall and splashes around into a pair of helices that collide with each other and create turbulence. By designing a helical inlet, we allow one of those channels of flow to dominate, and we minimize the turbulent flow that leads to platelet and blood cell damage. And we opened out the radius of our outlet side just a little bit to minimize the stagnation at the outlet cannula. And when we use computational fluid dynamics to measure fluid shear stress there's areas that aren't 100% perfect, but they're a lot smaller. And there's some edge effects from where the model, the computation runs up to the boundary of the modeled channel. And so, encouraged by our computational fluid dynamic results, we went ahead and began implanting these cartridges into experimental animals. And we've now gotten to the point where we can routinely implant silicon membrane nanofilter into an experimental animal for weeks on end, generate this watery ultrafiltrative plasma that we would describe as a primary urine waiting to be processed by tubule cells. And we're able to do this without any need for warfarin or heparin or any systemic anticoagulants. So I've shown you in a couple of slides how we've taken a concept of importing a new technology into the space from the microelectronics industry into renal failure that allows us to copy the advantageous aspects of nature's design of the kidney filters and successfully replicate that function in vivo. No pumps other than the animal's heart itself. So let's turn our attention now to the other half of the equation, the cells. If we're going to rely on living cells to reabsorb filtered salt and water and concentrate waste, we need the chemical energy of ATP. We need the proteins on the cell membrane that select what solute, salt, water, sugar, amino acids to take back into the cell. And we may need this elaborate subcellular structure on the top of the cell that expands the surface area of the cell and interposes these apical proteins throughout the fluid flow in the tubule. At the same time, the cells need to be able to block reabsorption and back leak of waste, and that refers to the tight junctions between adjacent cells. And here's where my cape-wearing optimism hit a barrier. This is a renal tubule cell from a healthy biopsy specimen. This image is from Hepton-Stahl's pathology of the kidney. You can see the top of the cell that's in the filtrate has this elaborate brush border, this kelp forest of fingers that place the reabsorbing channels all throughout filtered tubular fluid. The cells are packed with mitochondria, the energy plants, the power plants of the cell that produce the ATP, that allows channels in the basolateral surface of the cell to pump salt water. The moment that you take this cell out of the body and put it on a lab dish, all of these features atrophy. That lush kelp forest becomes this kind of peach fuzz. It's hard to find any mitochondria here. And so we were challenged from very early on that our project was actually doomed to failure because we were told that you could not grow cells on an artificial material and have them function with fidelity to the in vivo counterpart. And when I started asking people, how does this work? What goes on here? I got instructed to think about developmental biology and the interplay of protein and programs in a developing cell that lead it to take its final adult form. I have to tell you, it made my brain shrink. I just don't think this way. I lack this kind of biological thought process because in my heart, in my head, I'm a physicist and I'm an engineer. So I turned that barrier into an advantage. All I did is I simply wrote down what's the difference between a dish and an organ. How is the cell culture dish different from a healthy organ inside a healthy person? And it turns out it's a very long list. Organs are floppy and cell culture dishes are made out of hard plastic. Cells in the body have fluid flowing over them constantly, whereas cells in a lab dish, the fluid just sits there still. We worry about exhaustion of the cell culture media between media changes, and so we overload it with signaling molecules like insulin and food like glucose. So I just started going down the list, examining the impact of each one of these differences. And one of the first things that we found was that the elasticity of the scaffold on which we grew the cells matters. And so this was originally done by Ming-Pang Ao and then followed up by Hal Love in my lab, where he showed that if we grow the cells on a stiff substrate, we get these flat sheets of cell. And we all grew up thinking that this is good cell culture because you can see all of the tight junctions in green. You can see each nucleus one by one in blue. But I recognize that there's no structure in me that has this flat sheet shape. But when we grew cells on floppy substrates that approximate the same stiffness as a healthy human kidney, all of a sudden the cells start forming little cysts, little tubules growing on top of the material that look a lot more like what happens inside the human body. So these are just pretty pictures. They don't mean anything. Does this difference have any impact on the genetic programs of the cell and how they're activated? And this is where Rachel Evans saved the day, because we grew these cells on plastic dishes, on stiff substrates, and on soft substrates. And at first, you can see that we saw no difference in expression of a key transporter protein that's responsible in a healthy kidney for fluid reabsorption in the tubule. But Rachel Evans kept her copy experiment, her secret duplicate experiment, and ran it even longer, for weeks. Typical cell culture might be 10 days. Nobody grows cells out for a month. Rachel grew these cells out for a month, and after a month, all of a sudden, the expression of the key transporter protein came up, but only on the soft substrates. And it came up at the messenger RNA level, it came up at the protein level, and you could actually see it with indirect immunofluorescence microscopy in the lab dish. Okay, so that's a step forward. You can show that we can change the behavior of the cells based on the stiffness of the substrate. And you'll notice that this was done in 2018, 4 years ago. And at that point, progress suddenly slowed, because one of the challenges is that these soft substrates are made out of polyacrylamide, and they're not very water permeable. If we want to measure water transport by these cells, we can't do it on these substrates that we just used for testing because they're not water permeable. So we spent literally years probing different ways to try to grow these cells on permeable substrates that were elastic, and we got nowhere. So finally, I changed directions. I said, okay, we know that if gene expression in these cells is changing in response to substrate elasticity, then that must be happening because there's some signaling going on. Is there a known regulatory pathway that transmits information about the stiffness of the scaffold to the inside of the cell? And once I asked that, the answers started rolling. And in fact, I apologize that the bottom of this is cut off. But when we exposed these cells to an inhibitor of a specific gene regulatory pathway called the TGF-beta pathway, we saw that fluid transport on conventional cell culture substrates, the same substrates that I showed you before in the slide comparing the cell in the body and the cell outside the body. All of a sudden, transport went from 10 to 130. So that showed us that we could maybe copy some of the effects of the underlying tissue or the underlying plastic scaffold, not through changing what we grew the cells on, but how we controlled different pathways in the cell. And we went on to show that this was true with a variety of different cell types, that we could block the transport with a diuretic called tenapanor, showing that this was specific to the one transporter that we cared about, NHE3. We went on to show that this was also true for sugar transport, that when we blocked TGF-beta signaling, the gene for SGLT2, the sodium glucose cotransporter, went up both at the message level, at the protein level, and that when we measured sugar on the top side of the cells, it dropped dramatically in cells that got the TGF-beta inhibitor, and we could block it with a drug called fluorizin. That's an SGLT2 inhibitor. And finally, we asked the question about, could we use these cells to excrete waste? And the kind of classic model that we learn about in medical school for kidney physiology is a transporter called OAT1 and a chemical called paraminohippurate. And so we showed again that the expression of the transporters went up when we blocked TGF-beta signaling. We showed that excretion of paraminohippurate skyrocketed when we blocked TGF-beta, and that we could block this response when we gave a classic drug called probenecid. So now I've shown you that we've made a hemofilter that could be powered entirely by human heart, and we've made a progress in culturing cells for a bioreactor of living cells that can pump salt water and excrete toxins just using chemical energy. Now, there's one detail that I kept secret until now, and that is that these little gray picket fences here represent that silicon nanopore membrane that I showed you in the hemofilter. But you will have noticed that they're also over here in the bioreactor. Why is that? What are they doing over here? Well, what we realized very quickly was that if you've invented a good hemofilter that lets salt water through, lets sugar through, but doesn't let amino acids, doesn't let albumin, doesn't let clotting factors, and doesn't let antibodies through, you've also created the ideal scaffold for a bioreactor, because this blocks communication of the cellular and molecular effectors of the innate and the acquired immune system between the host's blood and the allogeneic cells. So we asked the question, well, does this actually work? It's a pretty idea, but there's a lot of pretty ideas that never came to fruition. So we devised a bioreactor architecture with silicon nanopore membranes, and we grew these kidney cells on the silicon nanopore membranes, and we planted them in an experimental animal, relatively short period of time, 3 days, a week, but enough that the innate immune system of the animal could attack these cells. And lo and behold, the cells on the chips stayed alive despite being in continuous connection to the animal's blood. So this gave us optimism that we were onto something, and we proceeded to say, okay, can we get this whole thing to work, the hemofilter and the bioreactor? So we assembled our hemofilter and our bioreactor into a microscale artificial kidney. And we directed the ultrafiltrate from the bioreactor, from the hemofilter into the bioreactor to see whether the kidney cells in the bioreactor would in fact begin to concentrate waste. So here's our small scale artificial kidney at the time of implantation into the animal. You can see the connections to the animal's blood vessels here and the connections between the hemofilter and the bioreactor here. Here we are opening the graft, and you can see the blood flowing through the bioreactor, the blood flowing through the hemofilter, and we're about to see the fact that we could get, in fact, little drops of fluid, concentrated waste, emerging from the cannula. And so in a 3-day experiment, using the hemofilter, using a bioreactor of actually a cell line, LLCPK1 cells from pigs, the hemofilter was putting out a very tiny fraction of the clinical goal that we have, but still a real amount of fluid. And the bioreactor was reabsorbing 90% of it, just as we need to take that targeted 20 liters a day of filtration and turn it into 2 liters of concentrated waste, reabsorption of 90%. And the cells stayed alive in the animal. So there you have it. Our strategy was to focus on the essential, the components of kidney function that you can't replace with pills or injections or drugs. We chose to copy the kidney strategy of filtration and concentration because that lets us get away from heavy roller pumps with batteries and drive lines. Let us get away from the swimming pool volumes of dialysate and copy Mother Nature's 20 million years of research and development in how to accomplish this physiologic task. We set a goal dose that was achievable. And I showed you that we borrowed a technology from the microelectronics industry to build a new technology filter powered solely by the human heart, and that we're able to let it operate inside an experimental animal with no clotting and no fouling and no anticoagulants. We've now overcome the most smug criticism of our project that everybody knows that tubule cells don't work in a dish. Well, tubule cells had not previously worked in a dish because nobody had asked them to. It's not that I'm a genius and brought this off. It's just that I asked the tubule cells to do something that wasn't important for prior researchers to replicate. And we've now integrated these two components into a small-scale bioartificial kidney that works in an experimental animal. My aunt had polio as an infant, and she spent time in an iron lung and survived to live another 75 years. Iron lungs clearly saved countless lives in the 1940s and 1950s. But that's not how we treat polio today. Iron lungs are medical museum pieces. Hemodialysis keeps 600,000 Americans alive from day to day in the United States today. But I would like to put dialysis machines into a medical museum right alongside iron lungs. And I'm going to close with a quote from Willem Kolk, who was the first physician to successfully treat kidney failure with dialysis in 1943. Kolk said, "It's nearly impossible to obtain funding for something simple. Therefore, over the years, my laboratory has teetered along, always on the rim of bankruptcy, always having too many ideas and too little money to carry them out." Thank you.